Sexual reproduction in flowering plants. We enjoy gazing at the myriads of flowers. We swoon over the scents and the perfumes of flowers. They attract us with their rich colors. These are present as an aid to sexual reproduction. Flowers for us do not exist to be used for our own selfishness. Sexual reproduction is shown by all flowering plants. A look at the inflorescences, their diversity of structures, flowers, and floral parts show a range of amazing adaptations that ensure formation of fruits and seeds, which are the end products of sexual reproduction. Flower, a fascinating organ of angiosperms. Human beings since time immemorial have had an intimate relationship with flowers. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. For conveying important human feelings such as love, affection, happiness, grief, mourning, etc., they have always been used as symbols. List at least 5 flowers of ornamental value that are commonly cultivated in homes and in gardens. Flowers, to a biologist, are the sites of sexual reproduction and morphological and embryological marvels. In class 11, you have read the various parts of a flower, which will help you recall the parts of a typical flower. Pre-fertilization, structures and events. The decision that the plant is going to flower has taken place much before the actual flower is seen on a plant. The differentiation and further development of the floral primordium resulted due to the initiation of many hormonal and structural changes. Floral buds and then the flowers are born in the inflorescences. The male and female reproductive structures, the androecium and the gynoecium in the flower, differentiate and develop. The male reproductive organ, the androecium, consists of a whorl of stamens, and the female reproductive organ is represented by the gynoecium. Stamen, microsporangium and pollen grain. Filament, the long and slender stalk, and the terminal, generally bilobed structure called the anther, form the two parts of a typical stamen. To the thalamus or the petal of the flower, the proximal end of the filament is attached. In flowers of different species, the number and length of the stamen are variable, each from different species. If you were to collect a stamen from 10 flowers and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Under a dissecting microscope, careful observation of each stamen and making neat diagrams would elucidate in different flowers the range in shape and attachment of anthers. The anther is bilobed in a typical angiosperm, with each lobe having two theca, that is, they are dithecous. Separating the theca, often a longitudinal groove runs lengthwise. In the transverse section of an anther, let us understand the various types of tissues and their organization. In the transverse section of the anther, the bilobed nature of an anther is very distinct. A four-sided tetragonal structure, the anther consists of 4 microsporangia located at the corners, 2 in each lobe. Further, the microsporangia develop and become pollen sacs. All through the length of an anther, they extend longitudinally and are packed with pollen grains. Structure of microsporangium. A typical microsporangium in a transverse section appears near circular in outline. It is generally surrounded by the epidermis, endothelium, middle layers, and the tapetum, which form the 4 wall layers. The function of protection and dehiscence of anther to release the pollen are performed by the outer 3 wall layers. The tapetum nourishes the developing pollen grains and forms the innermost wall layer. Dense cytoplasm and generally more than one nucleus is present in the cells of the tapetum. Can you think of how tapetal cells could become binucleate? When the anther is young, the center of each microsporangium is occupied by a group of compactly arranged homogeneous cells called the sporogenous tissue. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue to form microspore tetrads undergo meiotic divisions. What would be the ploidy of the cells of the tetrad? In the sporogenous tissue, each cell is capable of giving rise to a microspore tetrad. Each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. As they are formed, the microspores are arranged in a cluster of four cells, the microspore tetrad. The microspores dissociate from each other as the anther mature and dehydrate and develop into pollen grains. Several thousands of microspores or pollen grains are formed inside each microsporangium that are released with the dehiscence of anther. Pollen grain. The male gametophytes are represented by the pollen grains. You would find deposition of yellowish powdery pollen grains on your fingers if you touch the opened anthers of hibiscus or any other flower. On a drop of water taken on a glass slide, sprinkle these grains and observe under a microscope. You will really be amazed seeing on the pollen grains the variety of architecture, sizes, shapes, colors, designs from different species. Generally, pollen grains are spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent two-layered wall. One of the most resistant organic material known is the hard outer layer called the exine, which is made up of sporopollenin. It can withstand high temperatures and strong acids and alkali. So far, no enzyme that degrades sporopollenin is known. Prominent apertures called germ pores are present in pollen grain exine where sporopollenin is absent. Pollen grains, because of the presence of sporopollenin, are well preserved as fossils. A fascinating array of patterns and designs is exhibited by the exine. Why do you think the exine should be hard? What is the function of germ pore? The intine forms the inner wall of the pollen grain. The intine is made up of cellulose and pectin, which forms a thin continuous layer. A plasma membrane surrounds the cytoplasm of pollen grain. The vegetative cell and generative cell are the two cells present when the pollen grain is mature. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. A small generative cell floats in the cytoplasm of the vegetative cell. With dense cytoplasm and a nucleus, the generative cell is spindle-shaped. Pollen grains in over 60% of angiosperms are shed at this two-cell stage. In the remaining species, before pollen grains are shed, the generative cell divides mitotically to give rise to two male gametes. In some people, pollen grains of many species cause severe allergies and bronchial afflictions, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that a contaminant Parthenium or carrot grass has come into India with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. In recent years, it has become a fashion to use pollen tablets as food supplements. In the market in Western countries, a large number of pollen products in the form of tablets and syrups are available. The performance of athletes and race horses has been claimed to increase with pollen consumption. When once the pollen grains are shed and if they have to bring about fertilization, they have to land on the stigma before they lose their viability. How long do you think the pollen grains retain viability? Pollen grains, the period for which they remain viable is highly variable and depends on the prevailing temperature and humidity to some extent. In some members of Rosaceae, Leguminosae, and Solanaceae, they maintain viability for months, whereas in some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. You may have heard that for artificial insemination, semen or sperms of many animals, including humans, are stored. It is possible to store in liquid nitrogen the pollen grains of a large number of species for years. Such stored pollen in crop breeding programs can be used as pollen banks similar to seed banks. The pistil, megasporangium and embryo sac. The female reproductive part of the flower is represented by the gynoecium. The stigma, style and ovary are the three parts of each pistil. The pollen grains, the stigma, serves as a landing platform. The gynoecium may have more than one pistil and may consist of single pistil. The pistils may be fused together or may be free when there are more than one pistils. Beneath the stigma, the elongated slender part is the style. The ovary is a basal bulged part of the pistil. The ovarian cavity is inside the ovary. Inside the ovarian cavity, the placenta is located. The megasporangia, commonly called ovules, arise from the placenta. In an ovary, the number of ovules may be one to many. The megasporangium, a small structure, the ovule is attached to the placenta by means of a stalk called funicle. In the region called hilum, the body of the ovule fuses with funicle. Thus, the junction between the ovule and funicle is represented by the hilum. One or two protective envelopes called integuments are present in each ovule. The nucellus is encircled by the integuments except at the tip where the micropyle, a small opening, is organized. The chalaza, which is present opposite the micropylar end, represents the basal part of the ovule. The mass of cells called the nucellus is enclosed within the integuments. Abundant reserve food material is present in the cells of the nucellus. The embryo sac or female gametophyte is located in the nucellus. Formed from the megaspore, generally the ovule has a single embryo sac. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Generally, a single megaspore mother cell is differentiated by the ovules in the micropylar region of the nucellus. It is a large cell with a prominent nucleus and dense cytoplasm. The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Four megaspores are produced by meiosis. Female gametophyte. One of the megaspores is functional while the other three degenerate in a majority of flowering plants. The female gametophyte develops only from the functional megaspore. From a single megaspore, this method of embryo sac formation is termed monosporic development. What will be the ploidy of the cells if the nucellus, MMC and functional megaspore and female gametophyte? The functional megaspore nucleus forms two nuclei by mitotic divisions that move to the opposite poles, forming a two-nucleate embryo sac. By two more sequential mitotic nuclear divisions, four nucleate and later the eight nucleate stages of the embryo sac are formed. These mitotic divisions is of interest to note that they are strictly free nuclear, that is, cell wall formation does not take place immediately after nuclear divisions. The cell walls are laid down after the 8 nucleate stage, leading to the organization of an embryo sac or a typical female gametophyte. Inside the embryo sac, observe the distribution of cells. The cell wall surrounds 6 of the 8 nuclei and they are organized into cells. The remaining 2 nuclei, called polar nuclei, in the large central cell are situated below the egg apparatus. Within the embryo sac, there is a characteristic distribution of the cells. At the micropylar end, 3 cells are grouped together and constitute the egg apparatus. In turn, the egg apparatus consists of 2 synergids and 1 egg cell. At the micropylar tip, the synergids have special cellular thickenings called the filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. At the chalazal end, three cells are present and are called the antipodals. As mentioned earlier, the large central cell has two polar nuclei. Thus, at maturity, a typical angiosperm embryo sac, though 8 nucleate, is 7-celled. Pollination. You have heard in the preceding sessions that in flowering plants the pollen grain and embryo sac produce the male and female gametes respectively. For fertilization to occur, both the gametes have to be brought together as both types of gametes are non-motile. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains to the stigma of a pistil is termed pollination. An amazing array of adaptations are evolved by flowering plants to achieve pollination. To achieve pollination, they make use of external agents. Kinds of pollination. Pollination can be divided into three types depending on the source of pollen. Autogamy. In this type, within the same flower, pollination is achieved. From the anther to the stigma of the same flower, transfer of pollen grains take place. Complete autogamy is rather rare in a normal flower which opens and exposes the anthers and the stigma. In such flowers, autogamy requires synchrony in stigma receptivity and pollen release, and also for self-pollination to occur, the stigma and anthers should lie close to each other. Two types of flowers are produced by some plants such as Viola oxalis and commelina, flowers that are similar to flowers of other species with exposed anthers and stigma, called chasmogamous flowers, and flowers which do not open at all, called cleistogamous flowers. The anthers and stigma in such flowers lie close to each other. In the flower buds, when anthers dehisce, pollen grains come in contact with the stigma to effect pollination. Thus, as there is no chance of cross pollen landing on the stigma, cleistogamous flowers are invariably autogamous. Cleistogamous flowers, even in the absence of pollinators, produce assured seed set. Do you think that to the plant cleistogamy is advantageous or disadvantageous? Why? Geitonogamy. In this, the pollen grains are transferred from the anther to the stigma of another flower of the same plant. Although geitonogamy genetically is similar to autogamy, since the pollen grains come from the same plant and functionally it is cross-pollination involving a pollinating agent. 3. Xenogamy. In this, the pollen grains are transferred from anther to the stigma of a different plant. This is the only type of pollination which brings genetically different types of pollen grains to the stigma during pollination. Agents of pollination. Plants to achieve pollination use two abiotic and one biotic agent. Majority of plants for pollination use biotic agents. Abiotic agents are used by only a small proportion of plants. In both wind and water pollination, pollen grains coming in contact with the stigma is a chance factor. When compared to the number of ovules available for pollination, the flowers produce enormous amounts of pollen to compensate for this uncertainties and associated loss of pollen grains. Among abiotic pollinations, more common is the pollination by wind. Wind pollination also requires that the pollen grains are non-sticky and light so that they can be transported in wind currents. They often consist of well-exposed stamens so that the pollens are easily dispersed into wind currents, and stigma large, often feathery, is to easily trap airborne pollen grains. Often, wind-pollinated flowers in each ovary have a single ovule and numerous flowers packed into an inflorescence. The corn cob is a familiar example. The tassels are the stigma and style, which wave in the wind to trap pollen grains. In grasses, wind pollination is quite common. In flowering plants, pollination by water is quite rare and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall that among the lower plant groups such as algae, bryophytes and pteridophytes, water is a regular mode of transport for the male gametes. It is believed that for the transport of male gametes and fertilization, some bryophytes and pteridophytes distribution is limited because of the need for water. Vallisneria and hydrilla are some examples of water pollinated plants which grow in fresh water, and several marine sea grasses such as zostera. Water for pollination is not used by all aquatic plants. In a majority of aquatic plants, example water hyacinth and water lily, above the level of water the flowers emerge and insects or wind pollinate them as in most of the land plants. The female flowers in Vallisneria by the long stalk reach the surface of water, and the male flowers or pollen grains are released onto the surface of water. By water currents they are carried passively. Eventually some of them reach the female flowers and the stigma. In another group of water pollinated plants, example sea grasses, inside the water these female flowers remain submerged and the pollen grains are released inside the water. Pollen grains are long, ribbon-like in many such species, and passively they are carried inside the water. Some of them achieve pollination by reaching the stigma. Pollen grains in most of the water-pollinated species are protected by wetting by a mucilaginous covering. Flowers that are pollinated by both wind and water do not produce nectar and are not very colorful. What would be the reason for this? A range of animals as pollinating agents are used by majority of flowering plants. The common pollinating agents are bees, butterflies, flies, beetles, wasps, ants, moths, birds, and bats. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. In some species, even larger animals such as some primates, arboreal rodents, or even reptiles have also been reported as pollinators. Often flowers of animal pollinated plants are adapted specifically for a particular species of animal. Majority of flowers pollinated by insects are large, fragrant, colorful, and rich in nectar. A number of flowers are clustered into an inflorescence when the flowers are small to make them conspicuous. By color and or fragrance, animals are attracted to flowers. The flowers pollinated by flies and beetles to attract them secrete foul odor. The flowers to sustain animal visits have to provide rewards to the animals. The usual floral rewards are nectar and pollen grains. The animal visitor for harvesting the reward from the flower comes in contact with the anthers and the stigma. In animal pollinated flowers, the pollen grains are generally sticky and the body of the animal gets a coating of these pollen grains. The animal brings about pollination when the animal carrying pollen on its body comes in contact with the stigma. Floral rewards in some species are in providing safe places to lay eggs for the animal. In an example is the tallest flower or Amorphophallus. A flower itself is about 6 feet in height. Between a species of moth and the plant yucca, a similar relationship exists where both species, moth and the plant, without each other cannot complete their life cycles. The flower of the plant yucca gets pollinated by the moth, and in turn the moth deposits its eggs in the locule of the ovary of the flower. Observe some flowers of the following plants: cucumber, mango, people, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try and find out which animals visit them and whether they could be pollinators. Patiently, you will have to observe the flowers at different times of the day over a few days. Whether there is any correlation in the characteristic of a flower to the animal that visits it, try to see. If any of the visitors come in contact with the anthers and the stigma, observe carefully, as only such visitors can bring about pollination. Many times, without bringing about pollination, many insects may consume pollen or the nectar. Floral visits of such kind are referred to as pollen or nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy your efforts. Outbreeding devices. Hermaphrodite flowers are produced by majority of flowering plants, and pollen grains of the same flower are likely to come in contact with the stigma. Inbreeding depression results due to continued self-pollination. Many devices are developed by flowering plants to encourage cross-pollination and to discourage self-pollination. Pollen release and stigma receptivity in some species are not synchronized. Either the stigma becomes receptive before the pollen is released or much before the release of the pollen, stigma becomes receptive. The anther and stigma of the same flower in some other species are placed at different positions so that the pollen cannot come in contact with the stigma. Autogamy is prevented by both these devices. To prevent inbreeding, the third device is self-incompatibility. This is a genetic mechanism, and self-pollen prevents from fertilizing the ovules by inhibiting pollen tube growth in the pistil or inhibiting pollen germination. The production of unisexual flowers is another device to prevent self-pollination. In both male and female flowers, example castor and maize, are present on the same plant, it prevents autogamy but not geitonogamy. In several species, example papaya, on different plants, male and female flowers are present. That is, each plant is either male or female plant. Both autogamy and geitonogamy are prevented by this condition. Pollen-pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Often on the stigma, pollen of the wrong type either from other species or from the same plant also land. The pollen, whether it is of a right type or of a wrong type, the pistil has the ability to recognize. The pistil accepts the pollen if it is of the right type and promotes post-pollination events that leads to fertilization. The pistil rejects the pollen if the pollen is of a wrong type by preventing pollen germination on the stigma or the pollen tube growth in the style. The result of a continuous dialogue between the pollen grain and the pistil is the ability of the pistil to recognize the pollen followed by its acceptance or rejection. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. Some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection the botanists in recent years have been able to identify. As mentioned earlier, the pollen grain following compatible pollination germinates on the stigma to produce a pollen tube through one of the germ pores. Into the pollen tube, the contents of the pollen grain move. Through the tissues of the stigma and style, pollen tube grows and reaches the ovary. In some plants, you will recall that pollen grains are shed at two-celled condition. In such plants, two male gametes are formed by the division of the generative cell during the growth of the pollen tube in the stigma. Pollen tubes carry the two male gametes from the beginning in plants which shed pollen in the three-celled condition. After reaching the ovary, the pollen tube through the micropyle enters the ovule and then through the filiform apparatus enters one of the synergids. It has been shown in many recent studies that at the micropylar part of the synergid, the filiform apparatus is present and it guides the entry of pollen tube. From pollen deposition on the stigma until pollen tubes enter the ovule, all these events are together referred to as pollen-pistil interaction. Pollen-pistil interaction, as pointed out earlier, is a dynamic process which involves pollen recognition followed by inhibition or promotion of the pollen. In this area, the knowledge gained by the plant breeder would help in manipulating pollen-pistil interaction to get desired hybrids even in incompatible pollinations. From flowers such as pea, chickpea, crotalaria, balsam, and vinca, pollen germination can be easily studied by dusting some pollen on a glass slide containing a drop of about 10% sugar solution. Observe the slide after about 15 to 30 minutes under the low power lens of the microscope. Pollen tubes coming out of the pollen grains can be seen. In the chapter on plant breeding, as you will learn that a breeder to produce commercially superior varieties societies is interested in crossing different species and often genera to combine desirable characters. One of the major approaches to crop improvement program is artificial hybridization. It is important to make sure that in such crossing experiments only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen. This is achieved by bagging and emasculation techniques. It is necessary for the removal of anthers from the flower buds before the anther dehisces using a pair of forceps if the female parent bears bisexual flowers, and this step is referred to as emasculation. With a bag of suitable size generally made up of butter paper, the emasculated flowers have to be covered to prevent its stigma with unwanted pollen contamination. This process is called bagging. Mature pollen grains collected from anthers of the male parent are dusted on the stigma. When the stigma is bagged, flower attains receptivity and the flowers are rebagged and the fruits allowed to develop. There is no need for emasculation if the female parent produces unisexual flowers. Before the flowers open, the female flower buds are bagged. Pollination is carried out using the desired pollen when the stigma becomes receptive and the flower rebagged. Double fertilization. The pollen tube releases the two male gametes into the cytoplasm of the synergid after entering one of the synergids. Syngamy is completed with one of the male gametes moves towards the egg cell and fuses with its nucleus. This results in the formation of the zygote, a diploid cell. Towards the two polar nuclei located in the central cell, the other male gamete moves and fuses with them to produce a triploid primary endosperm nucleus. It is termed triple fusion as this involves the fusion of three haploid nuclei. Since syngamy and triple fusion, the two types of fusions take place in an embryo sac, the phenomenon is termed double fertilization, an event unique to flowering plants. After triple fusion, the central cell becomes the primary endosperm cell and develops into the endosperm while the zygote develops into an embryo. Post-fertilization, structures and events. Events following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruit are collectively termed post-fertilization events. Endosperm. Why does endosperm development precede embryo development? A triploid endosperm tissue is formed by the repeated division of the primary endosperm cell for the nutrition of the developing embryo. The cells of this tissue are filled with reserve food material. The PEN undergoes successive nuclear divisions in the most common type of endosperm development to give rise to free nuclei. The endosperm development of this stage is called free nuclear endosperm. Cell wall formation occurs subsequently and the endosperm becomes cellular. Before cellularization, the number of free nuclei formed varies greatly. From tender coconut, the coconut water that you are familiar with is made up of thousands of nuclei, which is nothing but free nuclear endosperm, and the cellular endosperm is the surrounding white kernel. Before seed maturation, the endosperm may either be completely consumed by the developing embryo or it may persist in the mature seed, and during seed germination it can be used up. Look for the endosperm by splitting open some seeds of castor, peas, beans, groundnut, fruit of coconut in each case. In cereals such as wheat, rice and maize, find out whether the endosperm is persistent. Embryo. At the micropylar end of the embryo sac, where the zygote is situated, the embryo develops. Only after a certain amount of endosperm is formed, most zygotes divide. To the developing embryo, this is an adaptation to provide assured nutrition. In both monocotyledons and dicotyledons, the early stages of embryo development are similar, though the seeds differ greatly. The proembryo is formed from the zygote, which subsequently develops into a globular heart-shaped and a mature embryo. An embryonal axis and two cotyledons are present in a typical dicotyledonous embryo. The epicotyle is a portion of embryonal axis above the level of cotyledons which terminate with the plumule or stem tip. The hypocotyle is the cylindrical portion below the level of cotyledons that terminates at its lower end in the radicle or root tip. The root cap covers the root tip. Only one cotyledon is present in the embryos of monocotyledons. The cotyledon is called scutellum in the grass family that is situated towards one side of the embryonal axis. The embryonal axis at its lower end has the radicle and the root cap enclosed in an undifferentiated sheath called coleorhiza. The epicotyle is a portion of the embryonal axis above the level of attachment of scutellum. Enclosed in a hollow foliar structure, the coleoptile is the epicotyle that has a shoot apex and a few leaf primordia. Overnight, soak a few seeds in water such as wheat, maize, peas, chickpeas, and groundnut. Then split the seeds and observe the various parts of the embryo and the seed. Seed. The final product of sexual reproduction in angiosperms is the seed, which is often described as a fertilized ovule. Inside the fruit, seeds are formed. Seed coat, cotyledons, and an embryo axis is present in a typical seed. As in legumes, due to the storage of food reserves, the cotyledons of the embryo generally are thick, swollen, and simple structures. Mature seeds may be non-albuminous or ex-albuminous. No residual endosperm is present in non-albuminous seeds as it is completely consumed during embryo development. A part of endosperm is retained by the albuminous seed as it is not completely used up during embryo development. Occasionally, remnants of nucellus are also persistent in some seeds such as black pepper and beet. The perisperm is the residual persistent nucellus. As tough protective seed coats, integuments of ovules harden. In the seed coat, the micropyle remains as a small pore into the seed during germination. This facilitates entry of oxygen and water. Its water content is reduced as the seed matures and seeds become relatively dry. The embryo's general metabolic activity slows down. The embryo may enter a state of inactivity called dormancy, or they germinate if favorable conditions are available. The ovary develops into a fruit as ovules mature into seeds, that is, simultaneously the transformation of ovules into seeds and ovary into fruit proceeds. The wall of the fruit called pericarp is developed from the wall of the ovary. The fruits may be dry as in groundnut and mustard, etc., or may be fleshy as in guava, orange, mango, etc. Mechanisms for dispersal of seeds have been evolved by many fruits. As an earlier class, recall the classification of fruits and their dispersal mechanisms that you have studied. Is there any relationship between the number of seeds present in the fruit and number of ovules in an ovary? By the time the fruit develops from the ovary, in most plants, other floral parts degenerate and fall off. However, the thalamus also contributes to formation of fruit in a few species such as apple, strawberry, cashew, etc., and such fruits are called false fruits. However, most fruits are called true fruits as they develop only from the ovary. Although fruits are a result of fertilization in most of the species, however, there are a few species of fruits which develop without fertilization, and these kinds of fruits are called parthenocarpic fruits. Example, banana. Through the application of growth hormones, parthenocarpy can be induced and such fruits are seedless. Several advantages to angiosperms is referred by seeds. Firstly, since pollination and fertilization are reproductive processes that are independent of water, seed formation is more dependable. Also, seeds for dispersal to new habitats have better adaptive strategies which help the species to colonize in other areas. As the young seedlings have sufficient food reserves, they are nourished until they are capable of photosynthesis on their own. The young embryo is provided protection by the hard seed coat. They generate new genetic combinations, being products of sexual reproduction leading to variations. Seed is the basis of our agriculture. For storage of seeds, dehydration and dormancy of mature seeds are crucial, which can be used to raise crop in the next season and also as food throughout the year. Can you imagine agriculture with seeds which germinate straight away soon after formation and cannot be stored, or in the absence of seeds, after they are dispersed, how long do the seeds remain alive? This period again varies greatly. The seeds lose viability in a few species within a few months. Seeds of a large number of species live for several years. For hundreds of years, some seeds can remain alive. There are several records of viable seeds yet very old. The oldest is excavated from Arctic tundra, that is the lupine or the Lupinus arcticus. After an estimated record of 10,000 years of dormancy, the seed germinated and flowered. A viable seed is of the date palm Phoenix dactylifera, which is 2000 years old, a recent record that was discovered at King Herod's palace near the Dead Sea during the archaeological excavation. It would be worth attempting, after completing a brief account of sexual reproduction of flowering plants, to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. In an embryo sac, how many eggs are present? In an ovule, how many embryo sacs are present? In an ovary, how many ovules are present? In a typical flower, how many ovaries are present? On a tree, how many flowers are present? And so on. In which fruit of some plants can you think would contain very large number of seeds? One such category are orchid fruits, and each fruit contains thousands of tiny seeds. In fruits of some parasitic species, is the case. Example, Orobanche and Striga. Have you seen a tiny seed of ficus developed from the tiny seed? How large is the tree of ficus? Each ficus tree can produce how many billions of seeds? Over the years, can you imagine any other example in which such a tiny structure can produce such large biomass? Apomixis and polyembryony. In general, the products of fertilization are the seeds, but a few flowering plants, such as some species of asteraceae and grasses, have evolved a special mechanism to produce seeds without fertilization called apomixis. Without fertilization, what is fruit production called? A form of asexual reproduction that mimics sexual reproduction is apomixis. The apomictic seeds have several ways of development. The diploid egg cell in some species is formed without reduction division and without fertilization develops into an embryo. As in many citrus and mango varieties, more often surrounding the embryo sac, some of the nucellar cells start dividing, protrude into the embryo sac and develop into the embryos. In such species, many embryos are present in each ovule. Polyembryony is the occurrence of more than one embryo in a seed. Take out and squeeze some orange seeds. From each seed, observe different sizes and shapes of the embryos. In each seed, count the number of embryos. What would be the genetic nature of apomictic embryos? Can they be called clones? Several of our food and vegetable crops of hybrid varieties are being extensively cultivated. Tremendously, productivity has increased by the cultivation of hybrids. Every year, hybrid seeds have to be produced, which is one of the problems of hybrids. The plants in the progeny will segregate if the seeds collected from hybrids are sworn and they do not maintain hybrid characters. For the farmers, the cost of hybrid seeds is too expensive because the production of hybrid seeds is costly. There is no segregation of characters in the hybrid progeny if these hybrids are made into apomicts. The hybrid seeds can then be used by the farmers to raise new crop year after year, and every year he does not have to buy hybrid seeds. In hybrid seed industry, because of the importance of apomixis in many laboratories around the world, active research is going on to transfer apomictic genes into hybrid varieties and to understand the genetics of apomixis. Thank you so much for watching. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click the bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.